Welcome to this next part of our webinar series. My name is Astrid Gall and I'm an outreach officer for Ensembl. Today's topic will be comparative genomics. In this session, we're gonna look at the different applications of comparative genomics and then the species for which we have this data available. I will talk you through our gene tree pipeline and how we make homology predictions. And I will also explain about our whole genome alignments, which are either pairwise alignments or multiple alignments. So why would you be interested in comparative genomics analysis? Well, a lot of people are interested in this because they're just interested in evolution. This analysis allows you to find differences between genomes of different species. And it allows you also to derive gene functions based on the homology between species. And finally, it can help you to identify highly conserved regions in genomes, which are regions which tend to be very important in the biology. The comparative genomics views in Ensembl that we have are organized into two broad types. On the one hand, we have gene-based resources, which you can find in the Gene tab. And on the other hand, we have sequence-based resources, which you can find in the Location tab. In the Gene tab, you will have phylogenetic trees for both proteins as well as for non-coding RNAs. You can get information about homologies and protein families. In the Location tab, you can find the whole genome alignments. You can get information about ancestral sequences and ages of bases in different tracts. There are also tracks for conservation scores and constrained elements. And finally, there is information available on synteny. And synteny refers to the concept of genes being present in the same order in two different species. Okay, our comparative genomics analysis in Ensembl is done by taxa. If you work, for example, with a vertebrate, human, mouse, or so on, you would first and foremost compare your species of interest to all the other vertebrates in Ensembl Compara. If you work with a plant, you compare your plant of interest with other plants in Ensembl Plants. There is a second level of analysis here, and this is called pan-taxonomic compara. Here we take a representative subset of genomes from Ensembl metazoa, fungi, plants, protists, and bacteria. So from all the different divisions in Ensembl genomes, our non-vertebrate sites, and compare across those species. We need to use a subset because we can't do this analysis for all species that we have, and the species that we use are chosen to be representative of different clades. Where can you find these two different levels? This is an example for a plant, Arabidopsis thaliana. And if you look at this screenshot here, we are in the Gene tab. And on the left-hand side in the menu, we have one section for plant compara, where you can find alignments, gene trees, and homologs. And just below that, there's another section for pan-taxonomic compara, where we have gene trees and orthologs across different species sets for a wider range of taxa. If you happen to work on a mouse, there's a more detailed level available too. You have the Ensembl Compara and the pan-taxonomic compara, and in addition, there's an analysis that we run across rat and all mouse strains and species that we have in Ensembl. Similarly, we do this also now for our pig strains, and as of yesterday, for our different rice species that we have in Ensembl. So as I said, we have two different tree pipelines for protein trees and for non-coding RNA trees, and they are very similar. However, I will explain just an overview of the protein tree pipeline here. So for this pipeline, we use a representative translation of each gene from all species in Ensembl, and then we do HMM searches to classify these into families, or we use clustering algorithms such as BLAST or H-cluster. For each of these clusters, we then run a multiple alignment, and we derive a phylogenetic tree for each of the clusters based on this alignment. And this is done with tree-best. Once we have these trees, we reconcile them against the species trees from NCBI taxonomy. And in the final step, we use our trees to infer our orthologs and pairwise. Pairwise and orthologs are both homologs. This is the overarching term. Pairwise are defined as genes that emerge through duplication events. And in my little example here with a gene in chimpanzee, human, and mouse, there's one duplication event shown in red, and accordingly, the genes C1 and C2 and H1 and H2 are pairwise because they're both emerged from this duplication event here. On the other hand, we have the orthologs, which are genes that emerged through speciation events, which are shown in blue in this tree here, and accordingly CH and H1, for example, are orthologs. For both of these, we have furthermore one-to-one, one-to-many, as well as many-to-many relationships. One-to-one means that there is one gene in one species, which is orthologous to one gene in another species. One-to-many means that there is one gene in one species, here in the mouse, which is orthologous to two genes in another species, such as these two human genes here. And then we have many-to-many as well, and this is not shown on this slide. Now I'm going to talk a little bit about alignments. So our alignments are whole genome alignments, and they come in two different flavors, either pairwise alignments, where you compare two species, or multiple alignments, where you compare a set of species. You can use these to identify highly conserved regions in the genomes, and these regions, as I said earlier, are likely to be sequences that have evolved slowly and therefore likely to be important for certain functions. Accordingly, they are often found in coding regions, but we find them in non-coding regions as well. Alignments also allow you to spot problems with annotation in certain regions, so they are worth looking at if you find a gene which you think may not be annotated very well. And finally, the alignments can be used to define syntenic regions. In Ensembl, we use different methods for our alignments according to which species set we are interested in. We have pairwise alignments that are either done with BLAST-Z or LAST-Z, comparing two species with each other. And then we have multiple alignments for sets of different species. And for these, we use three different methods. And which method we use depends on how closely related the species are. This EPO analysis is well suited for species sets where the species are relatively closely related. And we have a set of alignments here available for more than 100 mammals, 27 primates, and other alignments that you can see here. For species that are not so closely related, we use Mercator-Peak alignments. This is the analysis that we use for our amniota, so mammals and birds. And finally, we also do a progressive cactus alignments for our mouse and related species. And there's a link to our documentation at the bottom of this slide, where you can look up your species of interest and can find out which of these alignments are available for your species. Okay, so if you want to find out more about Ensembl Comparative Genomics, please have a look at this publication here. I'm now going to do a live demo. And this live demo has two parts. In the first part, we will look at the BRCA2 gene to find gene trees and homologs. And in the second part, we will look at a certain region of the human genome, which is on chromosome 2, here shown in red with these coordinates, and find alignments and conserved regions in this region on human chromosome 2. And this is the region that contains the HOX-D cluster. So I'm going to leave my presentation now. I'm going to copy this region for later. And I will go into my browser. Yeah, I'm excited to say we are at Ensembl release 100 now. This was released yesterday, in fact. So you are one of the first people working with this. So I'm going to search for the human BRCA2 here. And as previously, just click on the top hit, the human gene, to go to this gene. I'm now in the Gene tab for the gene BRCA2, and my Comparative Genomics section in the menu is just here. And the first thing I will want to do is to look at the gene tree. So I click on the gene tree here. You can see on my gene tree page that gene trees have a stable identifier as well. They start with ENS and then GT for gene tree. And this is a picture of my gene tree. I'll make it a bit smaller. And this is the picture of my gene tree here on the left-hand side and the underlying protein alignment on the right-hand side as an overview. In my gene tree, the gene that I have searched for is highlighted in red, so the human BRCA2 gene here. And then you can refer to the legend to see what the colors of the nodes mean. So we have blue speciation nodes and red duplication nodes. Also in the tree, you can see these gray funnels. So the gray funnels are hiding other sequences behind them. If you click on a funnel and then say, expand this subtree in the pop-up menu, you will see which species were hidden behind this funnel and this cluster. So in this case, many rodents. You have more options to change the views of this tree here at the bottom of the page. On the right-hand side, as I said, these are the underlying alignments. We have them color-coded in two different ways. For the collapsed alignments, so this is where we have these gray funnels, we either have a white, a light green, or a dark green, which stands for the percent of aligned sequences at any given positions. And for the expanded alignments, we only have gap or aligned sequence, shown in white and light green. You can, of course, download this tree as a picture. For this, you can use this export this image button, and you get different options similar to the location tab for the formats and specifications of this picture. With the trees, you can also download them in a format that can be used for a bioinformatics downstream application. And for this, you use this icon here where it says download data from this image. And here you can see the different formats that we support, standard alignment formats, tree formats such as Newick, Nexus, OrthoXML, and so on. So you can pick what your program supports here and then use this file after you have downloaded it. Okay, I'll close this again now. And now we're going to have a look at the homologs. there are orthologs and paralogs. In this case here for BRCA2, you can see that the paralogs are grayed out. And this is because there are no paralogs for BRCA2. Therefore, what we will do is we will go into the orthologs now and look at the gene in other species. On the ortholog page, you have two tables. The top table here gives you an overview of the different species sets available with their numbers of species in them, and then how many of these species have orthologs and what type they are. So if we look at rodents, for example, we have 31 rodents, of which 26 have a 1-to-1 ortholog with a human BRCA2, 2 have a 1-to-many ortholog, and 3 don't have any ortholog at all. You can see what these are by clicking on the 3, and it takes you to the bottom of the page where the species without orthologs are listed. Okay, I'll go to the top again. You can see my table with the details of the orthologs here at the bottom of the page. Is a huge table. We have something like 258 species. So I can actually filter this table by selecting my species set of interest in the top table here. So if I select rodents, my table at the bottom here only includes rodents. So this table here is the table with the details about the orthologs and other species. It includes the species with their name, what the ortholog type is, what the actual orthologous gene is with their stable identifiers, and where they are. And then you have information about the identities, and these come as target identity, which is the percentage of the orthologous sequence matching the human sequence, and the query identity, which is the other way around. You can also have a look at some information about how confident we are about these ortholog predictions in these other columns here. The columns can be sorted as well by clicking on the header, for example, for the identities. So I'm going to look at one ortholog now in mouse. So I'm actually going to filter this table by typing the Latin name of mouse in here. And you can see there's only one ortholog between a human BRCA2 and mouse. It has this identifier here. And what I can do now is I can view an alignment between human and mouse for this gene. So I click on View Sequence Alignments here to do this. And I've got the option to either show a cDNA or a protein alignment. And I will go for protein here. So I click on Protein Alignment in the pop-up. There we are. So my protein alignment has loaded. It comes in a cluster W format, which is a commonly used format for protein alignments. This has 3 lines. In the top line, I've got my human protein. In the middle line, I've got my mouse protein. And in my third line, I've got some characters. And these characters represent how conserved the residues are. Where you have a space, they are completely different. Where you have a star, this means that they are identical. And where you have a colon or a dot, it means that they are semi-conserved. You can download your alignment using this button here. And yeah, going back to the orthologs, you can of course download those as a bigger set as well using this button here. Okay, so this was my first part of the demo. I'm now going back to my homepage by clicking on the Ensembl logo. And I will now search for this region on human chromosome 2 with the HOXD cluster. So this is the region. And I click Go, and it will take me directly to this region on human chromosome 2. Remember from the first webinar that on this page we have 3 pictures: chromosome, overview picture in the middle, and then a highly detailed customizable view here. It's loaded now. And this we will configure now to show some information about conservation and some alignments. You will remember that we do this by clicking on Configure this page in the menu on the left. And in this pop-up menu here, there's a whole section dedicated to comparative genomics here towards the bottom. Some tracks are already on, but I will add some more. So I will go into conservation regions first. And here I will select the conservation scores for my 103 mammals based on this EPO alignment. And these constrained elements are already pre-selected, so I leave that as it is. What I will also do is I will go into the pairwise alignments, which are generated by BLASTZ and LASTZ. Here in this menu item, BLASTZ and LASTZ alignments. And I will select a couple of species for which I want to see an alignment with the human HOXD cluster. And I will go for chicken and chimpanzee here. And switch these on in a normal view. Oh, that was the wrong one. Chimpanzee. And now I will close this menu. And my tracks will load onto my 3rd picture on this page. Okay, it should be there any moment. You can see this grab element track is on by default already. This is the one that was pre-selected. And now my GURB scores have loaded as well. So what these 2 tracks show is they show the conservation of the nucleotide at that position. The conservation scores here are positive where there is conservation at this position, and they're negative where there is no conservation. And these raw data scores were then used to infer the constrained elements, as they are called. So everywhere where we have a peak here reaching a certain cutoff, we have annotated a constrained element. And these are these dark brown blocks here. These constrained elements are called constrained elements because they are considered to be constrained by evolution. And if you look closely at the scores, the elements, and then where they fall in relation to our genes, you will notice that constrained elements with high GURB scores are found in protein coding parts of the genomes and line up with the exons of the protein coding parts of the exons of the HOXD genes here. In contrast, in regions where there is no gene annotated, such as here, we don't find a lot of conservation. So negative scores and now constrained elements there. Okay, so now I will scroll to the bottom of this picture and have a look at my alignment tracks. I have chosen to show chicken and chimpanzee, and this is aligned against the human genome, which we are looking at. And in these 2 tracks here in pink, the pink blocks mean that there is an alignment at this position, and the gaps mean that there is no alignment in this region. And you can see what we would expect, that the alignment is best between closely related species, namely chimpanzee and human, aligns everywhere. For chicken, we have a lot of gaps there. Okay, I will scroll back to the top and show you some more comparative genomics views. There's the section here in the menu on the left where all of these are summarized. And we will now look at some alignments between two species. You can do this in an image and you can do this in a text format as well. And I will show you the alignments in a text format. So I click on that. And what I need to do here is I need to select an alignment. This was actually selected, but I will take you to this menu now. So if you haven't selected anything yet, you can click on the button, select an alignment, and you get to a alignment selector that looks like this. And then you can decide what species set or what individual species you want to look at. And I will go for mouse. So this is a pairwise alignment with human. And then I click through rodents and rats and mice to my mouse reference genome, which is then shown on the right-hand side, and click apply. And then I get this table here, which shows me that my two genomes align with each other in 4 blocks, of which block 1 is the largest one. And for this one, the location for both species is also on chromosome 2. So we would think that this is the most reliable and relevant alignment block. Therefore, I will click on that one now. And that takes me to a page that shows me a preview of this alignment block for the first nucleotides here. And you can display the full alignment within the web browser, but it may be more useful to actually download it and then pass it in some kind of bioinformatics downstream application. Finally, if you prefer pictures over text formats, you can also look at this visually in the region comparison view, where I go now. Here again, you need to select your species or region. I will click on this button and I will again select mouse as a species. The selector looks very similar. So I click through to rodents, rats and mice, and then the mouse reference genome and click apply. Yeah, and then I get a comparative view between human and mouse on the 3 levels that you can see in the region detail view on the location tab. So at the top here, we have got the human and the mouse chromosome with the location where we are highlighted in red. Please note the difference there. And then we have an overview of 1 megabase for human and mouse. And here you can start to see that the gene annotation in these regions is different. If you look carefully, you will note that there is a HOXD gene in mouse, for example, which is on the reverse strand. And then finally, at the bottom of the page, we have a highly customizable view again for human and mouse in this case, showing us the details about the different transcripts in this cluster and how this region aligns between the 2 species. So again, here in pink, we have regions that align and they are connected by these green blocks, and white is just gaps. So there are additional regions in both the human and the mouse genome here. And yeah, you can see the differences in the gene annotation as well. You can configure this view separately for your 2 species by going to configure this page. And then at the top, selecting your species and then changing or adding tracks you are interested in. Okay, this was the end of the demo part 2. I'll now head back into my presentation. and wrap up and show you some further resources. The next webinar in this series will be next week, Tuesday, 4 p.m. British Summertime or BST. It will be Ben, and Ben will be talking about the Ensembl regulatory build, which is using data from different sources, including ENCODE, Roadmap, Epigenomics, and Blueprint, to predict the positions and activity of features involved in gene regulation, for example, promoters and enhancers, and you will learn about how this build works and how you can find these features. If you have questions today on this webinar, please continue to ask them in the chat box. We will continue to answer your questions. I will check this in a few moments as well. We would encourage you to do the exercises for this module before the next webinar. You can find them in the online course linked on the left-hand side, and we will also upload our videos so that you can watch it again from today's webinar, as well as these slides and the demo that I've shown. If you need help with the exercises, you can go directly to the solutions in the online course. You can also use our living document, which you can find under this link. This is a document that you can all edit, so please be mindful of this and make sure you don't accidentally change or delete anybody else's question. And finally, you can also email us at helpdesk@ensembl.org with your questions. There are more resources available online. Ensembl is part of EBI training. The EBI has a lot of different bioinformatics courses available there. Ensembl also has their own tutorial on our webpage. We have a YouTube channel you have heard about. We are also on Youku. You can email us with your questions, or you can join our public mailing lists. If you are on the announced mailing list, you get information about the releases and the new data that we plan for the next release. If you join the developers mailing list, you can discuss your programmatic access problems there with people internally and externally to Ensembl. You can follow us on social media as well. We have a Facebook account, we have Twitter accounts, and we write a blog. This blog is published at ensembl.info. We also write publications, and you can find the full list on our homepage under this link. There is a yearly update for Ensembl as well as Ensembl genomes in nucleic acid research. These are the main papers. Please do cite either those or the specific publications which I mentioned earlier. If you use our resource, it's important for us that we get these citations as well. And this is us last year during our retreat. We are about 70 people. And I would like to thank the entire Ensembl team, here listed with their names, as well as our funders. Our main funding comes from the Wellcome Trust, and there is co-funding from different funding agencies, including the European Union and Ensembl.